So that brings us to the organelles. Pretty basic, pretty interesting. We have a number of various different organelles that we're going to look at. There's a number of other stuff that's inside of the cell, guys, and inclusions is just a general term that's used for it. I don't really get caught up on this, to be honest. You're going to see glycogen. We're going to talk about glycogen in terms of the muscles, okay? Because that's where your greatest glycogen stores are going to be. Glycogen is stored in the liver, okay? It's also stored within skeletal muscles. Skeletal muscles require a hell of a lot of glycogen to do stuff. Well, so we'll talk about that there. Melanin is going to be the one melanin that we're worried about is going to be involved with pigmentation. So it's going to be involved with skin. So we'll talk about melanin granules when we get to skin, or if we talk about skin. There's triglycerides. Triglycerides are long-term energy storage, okay? We have a number of various different cells that will bring in some inclusion bodies involving triglycerides. So we'll talk about these in some various different locations, but for the most part, I'm more worried about organelles, where they're at, if they're in a specific location, like nucleus, it's always towards the center of the cell generally. What they do, what their function is, okay? Obviously, that's going to be something that we'll talk about. It's something that you should be able to tell me on an exam. Okay. And there's some cytoskeletal structures. Microtubules, actin, myosin, some various different things that help to move things around. So organelles and cytoskeletal structures, those are probably things that I generally ask more of from this section. Again, that little review box at the beginning of the chapter here with all the organelles and their function. Yes, I expect you to know those. When we look at the cytoskeletal structure, let's go here to the picture here. There's a fabulous picture of showing you some of the microtubules there that are kind of specially stained so you can see them. You know, when we're looking at them in anatomy lab and you're looking at it at, 40x or whatever it is, you don't really see a whole hell of a lot of stuff. You definitely don't see the microtubules scattered throughout there. But there is a ton of cytoskeletal structure. It helps makes up the shape of the cell. It helps to hold the integrity of that cell shape, whether it's squamous or columnar or cuboidal. It helps to move stuff around. And I don't have, I'll see if I can pull up that video during break and we can see some of these bits and pieces in action here. But these cytoskeletal structures, not only do they maintain the shape and integrity of it, but they are spanning from one end of the cell to the other end of the cell. And they allow for this little railway system, if you will, for moving things from one side of the cell to the other. It's how the mitochondria move around. They move around through the interactions of those microtubules, them and the microtubules interacting with one another, okay? They're involved with cell division. They make sure that all of the chromosomes line up in the middle and get pulled apart at their centromeres and get moved around. Aside from moving things from one side of the cell to the other, having this little highway system, they do help the interaction of various different motor proteins. We're going to talk about kinesins and dyneins when it comes to moving things up and down nervous tissue. Okay? Neurons can be very, very short. They can be very, very, very long. All of it requires nutrients. And if it doesn't get it, obviously the nerve can die. So moving things around is a vitally important thing, especially when it comes to cells within the body here. And I will pull up that video during the first break, to show you guys when we come back, because you could see how these things are interacting with one another. And it looks like these things are walking around. It's kind of creepy. Don't I don't want you to get caught up in all the various different types of lysosomes. I want you to know what they generally have. Well, they're filled with digestive enzymes. There it is. There it is. This has some partially digested contents in it, so it's going to have some partially active digestive enzymes within it as well. Lysosomes, what does some mean? Anybody?
So, no, not so.
Lysosome, so means body, and lyse means to break or to destroy. Okay? So these are going to be cellular bodies that have digestive enzymes within it, and it's generalized. Okay, it's nothing specific. It's not like a peroxisome where it has hydrogen peroxide, peroxidases, if you will. Lysosomes, depending upon the cell, depends on how many lysosomes that cell has. If it's involved with the immune system, they're going to be filled with a ton of them. Okay. If these are cells that are within the digestive tract, there's going to be generally a lot of them. If it's a cell, maybe muscular system or whatnot, or any other system, it'll have a few of them. So depending upon the cell and its location, depends upon where it's going to be at. Liver cell, obviously going to have a ton of them. Lysosomes are just cellular bodies with some generalized digestive enzyme that fuses with things to break them down, whether that be a foreign invader, whether that be bringing in food, macronutrients. Okay. There are a number of them, primary, secondary. This is me not caring. Okay. When they fuse with the whatever, internet's bad today. When they fuse with whatever, whether it be a foreign invader, whether it be bringing in food stuff, or whether it be an organelle that's not functioning properly within the cell, okay, it fuses with it, digestive enzymes get released, and we destroy whatever it is we just engulfed. That's what lysosomes do. Okay. So what does that tell you? Lysosomes are a cellular body that contains digestive enzymes. They can fuse with anything. It could be a foreign invader. It could be food. It could be an organelle that's not up to snuff. Okay. So lysosomes are involved with maintaining the functionality of these cells. If we have parts of a cell that aren't working properly, that's referred to as autophagy. If we have cells that have gotten to the end of their life cycle, apoptosis, programmed cell death. They release chemicals. Those chemicals start breaking down organelles within inside the cell, the nucleus. Nucleus is dead, the cell dies, and we just make new ones, okay? New cells, all right? So that's what lysosomes do, okay? Peroxisomes. Peroxisomes are cellular bodies that contain peroxidases. These are going to be very specific enzymes that help to catalyze a lot of various different reactions. Some of the first substances that they produce in this pathway, that peroxisomes produce in this pathway when they destroy stuff, is very harmful to the cells. Hydrogen peroxide. There are a number of catalases, secondary catalases within a peroxisome as well that helps to convert that hydrogen peroxide into stuff that's inert and that cannot damage the cell, and that is water and oxygen. Okay. Where do we see these? We see them a lot within the liver. What does the liver do? The liver is involved with detoxifying the blood. Liver is involved with breaking down nutrients. Liver is involved with recycling old defective red blood cells. Liver is going to be involved in getting rid of foreign invaders. So the liver does a lot of things. So it would stand to reason that they would have a lot of lysosomes and a lot of peroxisomes. And that's what they do. Okay. You don't see those a lot in the testes. They're not breaking down stuff like that. If you don't know what the mitochondria does, I really don't know how you've gotten through school. Everybody knows it is the powerhouse of the cell. It has its own fluid, right? Called the mitochondrial matrix. It has a number of folds on the inside. It's a double-layered membrane. The folds in that innermost membrane are referred to as the cristae. You should know those two words. I assume you do. The cristae are there for one thing, and that is to increase surface area. And that increase in surface area allows for more surface area for chemical reactions to help make energy. Has its own DNA, has its own ribosomes, yada, yada, yada. There it is. Mitochondria, powerhouse of the cell. Outermost membrane, innermost membrane, highly folded. Again, those folds are referred to as the cristae. This is where the electron transport chain is. The ETC, the electron transport chain. That's where we find all the parts and pieces to do that. Yep, own DNA. Kind of interesting there. I'm not going to ask. I kept it in there because I thought it was interesting. That there are a number of mutations that are known to help contribute to various different human diseases. I just find that fascinating because you don't think of a lot of mitochondrial issues when you think of disease and whatnot. I just find it fascinating there. It is something that's only derived from mom. So in terms of 23andMe or whatever, ancestry or whatever you want to do, you can trace back mitochondrial DNA for a pretty significant number of generations there because it is only derived from mom. So it's something that you can follow back pretty easily there. They reproduce on their own. They're not really directed to do anything. They just provide a buttload of energy for a cell. Okay. And that is obviously a technical term. If I asked you what the function of a ribosome is, you would tell me it's involved with protein synthesis. It has two subunits associated with it, a large and a small subunit. And messenger RNA brings the information to the ribosomes that are floating around in the cytoplasm. They can also be attached to endoplasmic reticulum as well, okay? And if that's the case, that that endoplasmic reticulum is referred to as rough endoplasmic reticulum. But whether they are freely floating around in the cytoplasm or attached to endoplasmic reticulum, they always do the same thing. They read messenger RNA and they translate that message and make a protein, stringing individual amino acids together, i.e. the protein being made, okay? These proteins can do whatever. Remember the function of the proteins, all of those functions, whatever. The protein can be utilized inside the cell, or we'll package it up and we will remove it from the cell for use in another location. Okay. And in that video, it shows that process. It's kind of interesting there. Endoplasmic reticulum, and I will mention this here because on the exam, you may want to do it as well. If you want to shorthand something on an exam, guys, when you're typing out your answers, spell it once, put whatever moniker you have. For me, endoplasmic reticulum, I just shorthand as ER. And if you want to do that, that's perfectly fine. You got to spell it out once for me, please, so I know that you know what you're talking about. Okay. So endoplasmic reticulum, it's just a membranous bunch of tubes strung together. This is unique, ER. It extends all the way from the nuclear membrane to the plasma membrane. So it extends all the way from the nucleus and the center aspect of it all the way out to the periphery, okay, or pretty damn close. We have rough ER and we have smooth ER, okay? Rough, smooth, okay? And for whatever reason, this is the first time I've ever seen it. They're referred to it as granular endoplasmic reticulum and agranular endoplasmic reticulum, okay? If I put this question on the exam or on a quiz, I'll try to put it on as rough or smooth, but just be aware that there is a slight little nuance to the name here. Granular because it has a rough appearance, okay? So I guess if you look at this image up here on image A, I guess if you're looking at all this right here, okay, you can see the little circular dots. Those are all ribosomes, okay? So it has a granular appearance. I don't know, I'm fishing. A, remember when A is in front of a word, it always means without. Okay. So without granules, so smooth, whatever. Granular or rough ER always functions in proteins. Always functions in proteins, okay? Making proteins, right? They call it protein modification because, sometimes when a protein gets made, some things get clipped off of it, some things get added to it. It's modified, okay? But we're still making protein. Smooth ER or agranular endoplasmic reticulum. Its functions I don't even have listed here because it depends on the cell. Okay? If this cell is in the testes, the smooth ER is going to be modified to make testosterone. Okay? It's going to be involved with lipid synthesis, fats, and that's what testosterone is, fat derived. If this is within cardiac muscle or skeletal muscle, it's going to be involved with being specified to store calcium. So the function for smooth ER actually really depends on its location. If you wanted to write a function there because you feel obligated to, perfectly fine. Lipid formation, detoxification, liver cell, store calcium. Okay. Skeletal muscle, cardiac muscle. So depending upon where this cell is, depends upon its slight functionality difference. So smooth ER has a lot of varied functionality, all right? And it just depends on where it is. And we'll talk about all this when we get into these various different locales. When we, and this does a good job of showing it right here. Golgi complex or Golgi apparatus, you can call it either one. I don't honestly care. I interchange in what I call them. The book interchanges and what it calls them to in some instances, in some chapters. So all it is a bunch of flattened pancakes. Okay. And here you can kind of see all these beautiful flattened pancakes right here. The Golgi complex or Golgi apparatus modifies stuff. Okay. It receives packages from the endoplasmic reticulum. Okay. Golgi complex, where the rough ER is generally protein synthesis, making proteins. Smooth ER is generally associated with lipid synthesis, making fats. The Golgi complex can really modify both. Okay. It can receive materials from the rough ER and the smooth ER. But again, the functionality of the lipids, okay, of the smooth ER depends upon the cell. So I really only have proteins here, okay. But these Golgi complexes, they can receive from both rough and smooth ER. Okay. We have a little vesicle. It has a cis side and a trans side. I don't really care about that. We have a side of the Golgi complex that you can see receives a little vesicle from the rough ER. And it just travels through all these membranous tubes here. Okay. As it travels through these membranous tubes, this protein is going to be fine-tuned. A number of proteins are not ready once they've been manufactured there in the rough ER. They have to be modified to become fully active. Sometimes I have to cut off a few things. Sometimes I need to add a few things to get these proteins to be ready to use. Okay. And that's what the Golgi complex does. It gets the proteins and lipids, but proteins predominantly, ready for use, either within the cell or use outside of the cell. And again, we package them up into these little bitty vesicles. Okay. And we can move them around. You can say a lysosome can go and attach to it, destroy it. And I can use those parts of that protein that I made, those building blocks, those amino acids, and I can use them for other things within the cell. Or here, I fuse it with the plasma membrane and exocytosis. Bye-bye. See you later. And we can secrete these things from the cell. When I say secretion, you think of getting rid of. It's being squirted out of the cell. It's going, in the fluid. It's bye-bye. Okay. But sometimes this secretion is, I'm going to get rid of these proteins from inside the cell and I'm going to embed them within that plasma membrane. When we have some kind of immune response, this is what we see a lot of. Cells will make a number of proteins. Those proteins get embedded in the plasma membrane, and they are markers that the cell is going to pick up. So they're receptors that the cell is now going to be giving off. The body sees those receptors and it says, oh crap, I have a foreign invader inside that cell. So now those white blood cells going to go to the party. They're going to click onto that little bitty receptor that's been secreted into the plasma membrane, and it's going to enter into the cell and mount an immune response. So we have a number of fantastic functionalities of this Golgi complex. Proteins is generally the only one we really talk about.